Good morning, good afternoon, whatever it may be. How's everyone doing? Eating, all right. So I voyaged over from the other side of the river on this lovely day, and I see the lab sent one representative here to make sure I don't try to allege anywhere in this talk that I actually do anything technical in the laboratory, because I have a reputation as a storyteller but not a laboratory technologist. And so what I'm going to try to talk to you guys a little bit about today is the story of how we as a laboratory have moved from the technology of the 1990s and 2000s, old-fashioned Sanger sequencing, to next-generation sequencing. I have about 90 slides because if you asked me, I would talk forever on this, but I know you guys don't want to listen to me forever on all this, so I'm probably going to skip and choose a few slides here and there depending on what people's interest is and what people's questions are at different points in the talk. All right. So, and again, I'm a genetic counselor in the molecular diagnostics lab in adult neurology. So I'm going to give the backstory, and I say if someone invites you to grand rounds, you're allowed to pick on them at least once during the talk, but I see Lisa Scamenti did not come. But really, some of this story had its genesis in a conversation I had with Dr. Scamenti probably about 4 years ago. And the conversation we had was that she and some of her collaborators had identified a gene for a very rare skeletal disorder called Robinow syndrome. And the gene for the recessive form of Robinow had already been discovered. No one knew where the dominant gene was, and they had found it. And so she came to talk to me to say, hey, you work in the clinical molecular diagnostics laboratory. We've got this new gene. We think it would be great if you could bring this up as a diagnostic assay because no one else in the world does this. And, this was a pretty typical conversation we had with people at the University of Minnesota because that's essentially what we were in the business of doing in a clinical molecular diagnostics laboratory, of taking things that people had found here, gene-disease associations, developing robust clinical tests that we could then offer as a clinical service. This is what we did. And to show a little bit of our track record through the 2000s, so 2001 to 2010, we had taken 4 different genes that people here at the University of Minnesota had worked with. So the EMD, the LMNA gene for Emery-Dreifuss muscular dystrophy, the tyrosinase gene for oculocutaneous albinism, the PAX2 for renal coloboma syndrome. All of these are diseases that are very, very rare, but someone here at the University of Minnesota had made some connection between that gene and disease. And importantly, here at year 0, where we had the conversation with the researcher, nobody in the world offered diagnostic testing for that particular gene. And so it was a great opportunity for us to bring that up. And let's see if I can find the laser here. And what typically happened is once we brought up this testing very quickly, we got a lot of samples. And again, keep in mind, these are very, very rare diseases. So for example, for PAX2, there's about 180 cases in the whole world. And so to get 30 samples in a year is actually quite impressive. And so typically, things would move along very well like this, as long as we were the only laboratory in the Western Hemisphere doing the testing. The problem was that the curve didn't continue like this. What ended up happening was a laboratory that came before us in alphabetical order typically would develop a competing test, and literally almost within a month, our volume would drop to 0. And what this really was, it was an unsustainable way of us continuing to do business in the Mendelian disease arena to put 2 years, tens of thousands of dollars into developing and validating these rare sequencing disorders, only to have someone develop a test 2 years later and simply drop our volume to 0. And so Lisa brought it up to me, I brought it up to our lab director, and the answer was a quick no. And so from a business reality, from a survival standpoint, it wasn't something we could do to keep operating in this single gene model. In parallel, well, I guess I'm going to go and talk a little bit about, so backing up, that's a little bit of the backstory that started the conversation about why get into next generation sequencing. It sort of begs the question of why do we sequence genes? Why do we go through and determine the sequence of genes? And it's a good question because for some genes, like with factor V Leiden, there's one place in that gene that everybody who's got that mutation has the same mutation at the same position. And it's very easy to design a robust assay to look at that one spot and make the diagnosis. And so usually we prefer, if there's a single place in a gene, we prefer just to look there for the answer. The problem is for a lot of Mendelian diseases, there isn't one single place in the gene to look. There's thousands of places to look. And so the example, PKU, in the PAH gene, there's probably over 500 different known mutations. And so if you want to develop an effective diagnostic test for PKU, you need to be able to sequence across 1,300 or 1,400 base pairs of DNA and determine the sequence of that gene. And so when I say sequencing, what are we doing? Well, essentially, we're determining the sequence of nucleotides, A, C, T, and G, from beginning to end of both the alleles for autosomal genes, or for X-linked genes, there may only be one in a male. We are identifying any differences between the patient we're testing and the reference genome. And just because there's a difference doesn't necessarily mean that's pathogenic or it's a mutation. It may be a normal variation. So we're identifying differences, we're comparing it to the knowledge that we have, and determining, is this variant normal, benign, pathogenic, or increasingly we run into a lot of things that are unknown. And I couldn't put enough question marks around that, because that really is probably one of the biggest issues we run into. The bigger and bigger we scale up DNA sequencing, the more and more of these unknowns we get. So Sanger sequencing, which is essentially the DNA sequencing technology we used through the 1990s and 2000s, it's very robust. When I say we're leaving it behind, it isn't because it's not accurate. It isn't because it's not cost-effective at certain scales. It is a very accurate and robust way of looking at genes, but it's also very labor-intensive. And the reason is that for every single fragment of a gene, typically for every exon of a gene, you have to amplify that. You have to run sequencing reactions on the forward strand, the reverse strand. You have to analyze all that data exon by exon by exon, which is manageable in a 3 or 4 exon gene. But some of the genes we're talking about, our biggest one so far, has 373 exons. It becomes very cost ineffective at a large scale. And so this is an example of what Sanger sequencing data looks like. So you guys have probably seen this in the past. Essentially, we generate these colored peaks, and we look for positions where the patient is different from the reference. And so I talked about, well, why from a business standpoint it didn't make sense for us as a laboratory anymore to keep bringing up single gene tests only to be undercut 2 years later. In clinic, there were sort of parallel developments that really were making this model of genetic testing obsolete. And I use ataxia as an example because that is my primary clinical place where I work, is the ataxia clinic. And I think most of you guys know what ataxia is. It's a neurodegenerative disease. It's very genetically heterogeneous. There's many different types of ataxia. But for the point of this talk, I think what I want to point out is, in the beginning, the first few genes that were discovered in the 1990s, we explained about half of what ataxia was with 4 genes. And so it seemed like, gosh, if we find 3 or 4 more, we've got this thing solved. The problem is, it didn't work out that way. That increasingly, this unknown component is made of increasingly rarer and rarer things, to the point where if you want to offer an effective diagnostic test for ataxia, you need to be able to look at this point in time, dozens of genes, and I would argue probably in the next few years, hundreds of genes. Unless you think this is just ataxia, I could put up the same graph for hearing loss, retinitis pigmentosa, muscular dystrophy, the same things happening across all of these conditions where in order to have an effective diagnostic test, you need to be able to cost-effectively look at large amounts of DNA. And to do this by Sanger sequencing would cost hundreds of thousands of dollars. So we have sort of these twin pressures that led us to a point in March 2010. We sort of sat down and said, literally, is it time for us as a small academic diagnostic laboratory to give up on Mendelian rare diseases and just focus on some of the other things we do? We do a lot of other things for cancer diagnostics, bone marrow engraftment, and we're really at a point where the current model we had for Mendelian disease wasn't going to work going forward. But what we decided to do, thanks to I think a couple people who really decided we wanted to stay in this, and if we're going to stay in this, we needed to really go big, and we needed to try to get up to what is going on in DNA sequencing in 2010. And so we formed this collaboration we called the Clinical Genomics Core, which was composed of the pathologists and some of the technologists from our laboratory, the Minnesota Supercomputing Institute, and that's an important point because you start to generate so much data with this technology that there's no way you can analyze this without massive computing resources. The University of Minnesota Genomics Center, Kenny Beckman with the Genomics Center, he had all of the instruments and expertise. It's very expensive to get into this business. These instruments are incredibly expensive. The computing power is incredibly expensive. What we realized here was we had all these resources here. We just needed to talk to each other to figure out a way to apply this to clinical diagnostics. And I didn't really fit into any of the boxes, so I'm over there for genetic counseling services. But essentially, how do you take this information and give it back to patients? Make it meaningful for your patients. So what we're going to do is walk through what these different groups here do at the University of Minnesota to give you an idea of what next-generation sequencing is. So this is the University of Minnesota Genomics Center, and Kenny Beckman's the director of that. And when I talk about next-generation sequencing, I think people throw it around as if it's like a technique, it's like PCR, it's something else. Really, next-generation sequencing isn't so much a technique, but it's a way of analyzing DNA. And there's a lot of common elements, but there's many, many different subtleties and different platforms that next-generation sequencing can be done. The main point is what it allows you to do is look at massive amounts, genomes, if you will, in massive amounts of genetic information in a cost-effective manner. And the way this works is actually begins rather inelegantly. We actually just break up the genome into random fragments. And so this is typically, we do this with sonication. We're essentially just shattering the genome into hundreds of millions of random fragments. And then what we do is we take all of these fragments and we ligate adapters onto both ends. And what these adapters are, they're essentially molecular toolkits. So you have these hundreds of millions of unique fragments of genomic DNA from the patient's blood, but on the ends of all of them, there are the exact same adapters. And what these adapters have are things like common PCR primers. And so you can take the same PCR primers, the same sequencing primers, and you can manipulate all of these hundreds of millions of fragments with a single primer set. They also contain sequences that recognize a physical surface. So in the case, we use Illumina, and these adapters contain sequence that allow these fragments to go and bind to unique physical locations on a surface. So in other words, allow that fragment of genomic DNA to be physically isolated from all the other ones, so we can sequence that one fragment and determine its sequence. And the other thing that's important with these adapters is we can put in barcodes. And essentially, this technology is so powerful and gives us so much genetic information that we can actually take 4, 5, 6, 7, 8 people, mix their samples all together if they have a barcode in them, sequence them all in the same well, and bioinformatically later pull them apart based on which one of these barcodes we've given to their DNA. And so what's happening in this reaction essentially is we're taking all these random fragments of genomic DNA, we're putting toolkits on the end, and then we're going to start manipulating these. With Illumina, which is what we use, what happens is we take these fragments, we're going to bind them to this flow cell, and the surface of the flow cell is essentially coated with a lawn of nucleotides that are complementary to those adapters. So the idea is we take all of these hundreds of millions of fragments of genomic DNA, we wash them across this flow cell, and each one finds a unique spot, binds to these adapters, and lands there, and now it's physically linked to that spot, physically linked to a unique location on that slide. And these locations are unique enough that you can image that slide and tell all these different places apart. Once you've bound all those fragments to the slide, what we do then is we simultaneously sequence all of these hundreds of millions of fragments that are all physically bound to the slide. And I'm going to skip over a lot of details of this because I figure it's not the main point of today's talk. But in essence, what happens is with each strand, a single nucleotide is incorporated, and then the camera that's taking a picture records what color nucleotide was incorporated in that strand at that position. And this happens one base at a time, but you have to remember that there's hundreds of millions of fragments on this slide, all of which are glowing according to what's the next nucleotide put in place. So the power of this comes in the amount of DNA that you can sequence at the same time, which is why it's also called massively parallel sequencing. Essentially, you can simultaneously sequence hundreds of millions of fragments of DNA all at once. Okay? The other thing that I'm going to talk about a little bit is I may refer to what are called paired end reads or the different paired ends. What's happening there is when we take these genomic fragments of DNA, we put the adapters on the end, we're reading a small snippet of DNA this way and a small snippet of DNA that way. So a little bit over here, a little bit over there. We're not reading anything in the middle. There's a lot of reasons why, which could take another 20 minutes, and I won't go into. But essentially what we're doing is taking a fragment of DNA, we're reading a little bit here, a little bit here. We're ignoring everything else in the middle. But what we can deduce from this is, if we know that this is a 500 base pair fragment, if we map these reads to the genome, this read should map 500 bases away from that read. And if they don't, that may give us clues about structural rearrangements in the genome. Okay? So when the University of Minnesota Genomics Center is all done for our particular patient samples, we're generating 40 to 70 million of these paired end reads. So essentially 4 to 7 billion bases of DNA. And not only all those bases, but all the accompanying quality information. So this is a massive, massive data set, about 20 gigabytes or more of data per patient. And this is one of the biggest challenges with all this is, A, how do we store this? I'm pretty sure Fairview is going to notice pretty soon the hard drive's getting kind of full. But how do you manipulate this amount of data? This was beyond anything we had ever worked with in our laboratory. How do you go through this and find variants? And so this is where the Minnesota Supercomputing Institute came into the equation. And I remember we had met several times. We were all gung-ho, like, yeah, we're going to sequence exomes. We're going to do all this. And probably the most sobering conversation we had was when the bioinformaticists walked into the room. They're very, very nice people, but really started to point out how much infrastructure we needed to build in terms of informatics and how much that infrastructure really cost to get supercomputing resources that could handle this amount of data. But also, one of our unique needs was if we ran this once a month, we needed the infrastructure to handle this much data once a month, and then we didn't need it for 29 days. And then we would need a massive infrastructure again in about a month. And so that gets to how we solve some of these problems. We ended up building a cloud-based mapping and genotyping pipeline. And so essentially what we can do is rent some of the world's biggest supercomputers by the hour, upload these gigantic files, map all these reads onto the reference human genome, make all the calls, pull the data back down, kill the instance, and then we don't own a supercomputer anymore. And it really was sort of an ingenious way of doing this that was much more cost-effective than us having to buy all these computing resources. And to sort of walk through what happens once we receive a sample in our laboratory, we actually de-identify the sample, give it a number that only we know how to connect back to the patient, and then we release that to the University of Minnesota Genomics Center. As part of this process, we took Kenny Beckman's lab through the CLIA certification process, so he is now a CLIA certified laboratory able to generate this data, but he gets no patient identifiers in his laboratory. That data set is uploaded to the cloud. All of the reads, all of these 40 to 70 million reads we're generating are mapped onto the reference genome. Calls are made. We bring all that data back down into our laboratory, re-identify it, and make the interpretation of the data. And so it's a pretty slick way of doing this that we could not have done without this bioinformatics resource. I'm going to skip slides here and there, like I said, just because I don't think they're important. So what ends up happening is every one of these reads that we've got gets mapped to the human genome somewhere. We know what the human genome is. We've got a reference genome. And so any of these reads that we generate as part of the next generation sequencing get mapped onto the genome, and they get assigned a mapping quality score. And that's based on a couple of factors. One is, well, if I think it belongs here, is there anything making me think that it doesn't match perfectly? So in other words, it maps here, but there's a couple places where it really doesn't match the reference sequence. And so those would count against the mapping quality. So essentially, the better your reference sequence maps and matches, the better your patient sequence matches the reference, the higher the mapping score. And importantly, if there's other places in the genome that this read could map, that also reduces your mapping quality. And this is important because about 20% of our genes have pseudogenes in the genome. And we need to be aware that we may be generating sequence from a pseudogene and trying to throw it onto the real gene. And so there's a lot of important data that comes from this mapping quality of how confident am I that this came from here, and how confident am I that it could not have come from anywhere else in the genome. And so this is just a visual picture of what this data looks like. This is the SIGN1 gene that, of course, is associated with the type of ataxia I work with. And this is just the reference sequence, and this is a picture of thousands of these paired end reads all mapped across that gene. So this is kind of visually what this looks like when the data is mapped. We then go and we essentially look at every single position that we're analyzing, and we look at, of all the reads that have landed at that position, how many of them have the normal, the reference genotype, how many of them have a mutant or alternative genotype, and based on that, we make a determination of, is the patient normal, 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 mutant, or mutant, mutant? Are they heterozygous, homozygous, homozygous, normal, based on that? So this is an example of a position where half the reads at this position have a T. The other half don't have anything, which just means they're normal A. This is a patient who's heterozygous TA. And you can see, whenever you're generating billions of bases, there's a lone G hanging out up there. There's plenty of error and background noise that goes on with this. But what you're really looking for is a consistent signal that tells you this patient is homozygous at this position. I'm sorry, heterozygous at this position. And so the unelegant product, which I think the bioinformaticists would call this a thing of beauty, I don't think it's very beautiful, but this is what the end product is of that entire workflow. It's a massive spreadsheet of chromosomal positions, reference genotypes, patient genotypes, confidence scores. It's essentially a bunch of meaningless gibberish. And this is what exists after the bioinformatics portion of the pipeline. So when people say things like, oh yeah, you can get your genome for $1,000, well, you can get a million-line spreadsheet like that for $1,000. It doesn't tell you anything about what those variants are, what they mean for you medically. And this is where we come in. And the Molecular Diagnostics Lab, I did put you on there, Aaron, just so you know. All right. So we are in the business of taking this data and turning it into a meaningful report for patients. And this quote actually came from one of our very first meetings where the bioinformatics and the University of Minnesota Genomics Center were running away with all these great ideas. And I started talking about, okay, so I want to tell the patient, are they heterozygous, homozygous? And they were astounded that we actually wanted to tell an individual person what their genotype was from this data, just to show the different perspectives of what goes on in the research setting versus when you actually have to say to a patient, this DNA change is causing your disease, or this DNA change means you're going to get this disease. It's a very different world, and what we're in the business of doing in the clinical laboratory is turning this data into meaningful clinical information. So the first step is, well, we take the spreadsheet, we manipulate it to figure out what all those changes mean. And then what we're going to look at is a lot of data about all these different variants. Is it known? Have we seen it before? Have other people seen it before? If it's known, is the allele frequency consistent with a pathogenic role? In other words, is it rare enough that it could potentially be the cause of the disease I'm looking at? If I'm looking at a disease that affects 1 in 350,000 people, but the variant occurs in 50% of the population, it ain't the cause of that disease. And if it's novel, what can we do to predict the potential effects of this variant? And so we've got a lot of different resources we can use. We use locus-specific databases, which are voluntarily curated databases of variation, both normal and abnormal, in a lot of the genes we look at. We try to see, have people reported this particular variant as causative for the disease? Or have people consistently reported it as a normal variation? We use a lot of these big population databases that have been generated from the 1,000 Genomes Project, the 6,500 Exomes Project. And these databases are really most helpful to get a sense of, if you see a variant that's rare, is it really rare enough to be the cause of the disease you're looking at? For most Mendelian disorders, even something that's present in like 1 in 500 people may not be rare enough to be the cause of the disease you're looking at. And so if you really want to get a measure of how rare something is, you need to look at like 6,500 people, 13,000 exomes, to get a sense of how truly rare a variant may be. And to give you a sense of this, this is a variant that causes Fanconi anemia that has a minor allele frequency of less than 1 in 12,000. So in essence, we use these databases to try to get a sense of, is this variant rare enough to be the cause of the phenotype we're looking at? And there's all these imperfect in silico models, so you guys may see these or may hear about these things like polyphen, SIFT. Essentially, they're different ways of looking at different variations, whether they be amino acid substitutions, even silent silent mutations, intronic mutations, and trying to predict informatically, would we expect these to cause disease or not? None of these things individually tell us that a variant is pathogenic. It really is the weight of the evidence from all these different sources that leads us to say we think it's pathogenic, we think it's the cause of the disease, or we think this is benign, or We really don't know. Maybe we need to look at some other family members with the phenotype. Okay, so in the interest of time, we're going to skip. Lest people think that everything that comes out of next-generation sequencing is the truth, a lot of the work that we've done in the last year or two has been cataloging positions that are very prone to artifactual calls. And so Dr. Cashton brought up at some point here when I was talking to him earlier about, some labs still do Sanger sequencing of certain regions of genes. We do that too. We had to go through, we look at about 11,000 exons by next-generation sequencing. And of those, probably about 2 or 3%, so 300 or 400 exons, have very clearly problematic positions like this, where you start to get a whole bunch of garbage. And so we have to catalog, identify, and Sanger sequence or go to some other method to analyze these sections of the genome. So a lot of the work we've done to make this ready for clinical work is identifying these places and figuring out workarounds. So at this point, we're looking at about 11,000 exons. Probably 300 or 400 had some significant issue, which we've been able to work around in all, but I think 3 or 4 of those 11,000 exons. So those frustrate us and they stump us, but that's a pretty good success rate. And so this is showing how in this sequence you see all these different calls here, but when we go and we look at this by Sanger sequencing, the sequence looks clean and beautiful and those calls aren't there. So just going to show that next generation sequencing doesn't solve all these problems, but there are ways of working around those. So one of the big things we had to look at and try to figure out very early on was, okay, so we're going to go all in on this. We're going to jump onto this bandwagon and try to join next-generation sequencing. How big do we want to go? Do we want to just bring up maybe one clinical test like ataxia or hearing loss or something that's relevant to people at the University of Minnesota? How big a scale do we want to work at? And importantly, what metrics do we say are good enough for a clinical report? And this is important. This field is moving so quickly that there are no metrics. Nobody says, here's the level you have to sequence to be acceptable for clinical care, or to exclude mutations with this much confidence, or to identify mutations with this much confidence. It's moving too quickly that people have not established any of those parameters for clinical care. And so we had to figure out what are our metrics for good enough for clinical care. And this last one, how do you juggle or balance cost, trying to keep this cost effective, trying to maintain a reasonable sample volume, reasonable turnaround time. Each of those things often comes at the cost of the other. And how do we balance those variables? So to get to that, really to answer that question, we need to back up a little bit and talk about coverage. And when you hear people talk about next generation sequencing, you'll hear people say, oh yeah, we have this level of coverage or this much average coverage or minimum coverage. Coverage is a really, really important concept for answering those questions I was looking at. And what coverage means is, for any particular position in the genome, how many independent reads did you get across that position? In essence, how many times did you sample that patient's genotype at that position? The reason that's important is the more you sample a particular position, the more confident you are that you know their genotype. And if you think about a coin, if I had you flip it 3 times, would you bet $1 million? And you got heads all 3 times, would you bet $1 million that there's a tails on the other side? Or that it's heads on both sides? No, you really can't figure out. You really want to flip that coin 20 times. If you get 20 heads in a row, then you can start to be a little more sure, gosh, something's funny here. I'm getting heads over and over and over again. Same thing here. If we sequence across a gene, there may be positions where we get 1,000-fold coverage. We sample that position 1,000 different times, and we can be highly, highly confident in our genotype. If that patient's heterozygous at that position, we're going to have normal reads and abnormal reads there because we've sampled that genotype 1,000 different times. If we go over to here, we've only sampled that 4 times. And it's very plausible. They could be heterozygous for G and A, and if we only sampled 4 times, it's plausible we could have sampled the G 4 times and just missed the A by chance. So the deeper the coverage is at any particular position, the more confident we can be in making a genotype call at that position. Okay? Does that make sense? So what we have to try to figure out, and this was trying to illustrate the same point again here, just with smaller. what we had to try to figure out is, well, what is the minimum? If 4x isn't enough to make a confident call, what is enough? And there really was no standard out there. And so what we decided was that for every gene or any gene that is ordered by a clinician, we need to cover every single coding position within that gene 20x or more. And so if there's an exon that drops way down below 20x, we have to figure out another way of looking at that. Even if you have 1,000 base pair exon and one position drops to 19x, we go and we Sanger sequence that. We set that as our bar, that if we're testing a gene and either identifying or excluding mutations, we need to have looked at every single position in that gene 20 times minimum, or else we need to think of a workaround for it. And this has kept our techs very, very busy in the lab sequencing all these areas. So the challenge that brings up is essentially the cost of next-generation sequencing is related to how many reads or how much data you generate. So the more data you generate, the more reads you generate, the deeper your coverage, but the more expensive your test is. Okay? So if we look at the human genome, 97% of the human genome is non-coding DNA. I think probably most of us at this point would hesitate to call it junk. It probably has some very critical function. But it isn't something we currently analyze for a lot of diagnostic tests. Of the 3% that's coding, really only a small fraction of those genes are even associated with a human phenotype. And of those few thousand genes that are associated with a human phenotype, we probably only care about a few hundred of those here at the University of Minnesota. And so the problem this brings up is, if we just take genomic DNA and we break it up and we throw it all onto the sequencing instrument, start generating data, 99.9% of the data that we are generating maps to places in the genome we don't care about. And this was really the obstacle to adapting a lot of these technologies to clinical diagnostics. If someone said, I want to look at the PKU gene by next-generation sequencing, well, you have to sequence the entire genome and spend hundreds of thousands of dollars on this just to get the depth of coverage you need within the PKU gene. And so the way this problem was solved and the way that next-generation sequencing really was able to move into clinical diagnostics was the development of sequence capture methods. And so this is the package insert from Agilent, which I don't think clears up anything all that much. And so I made my own little graphic. And essentially what the idea is, you or we as a laboratory decide there are a few particular genes that we think are important for us to identify. We want to focus all of our sequencing capacity onto those genes and not waste it on all these other genes in the genome. And so for this, I just use the example. I use little 4 base pair snippets. Let's say we're interested in the sequence GTGT. That's the gene we're interested in. What we do is we design a 120 base pair biotinylated bait that binds to that sequence. And then we put that in with all of our genomic DNA. Those baits go up. They find the sequence that they align to. And then we can pull those down with streptavidin beads and magnet. And essentially what we do is we are pulling out these sequences that we're interested in analyzing and creating a pool of DNA fragments that is highly enriched for the things that we want to look for. I put that other one in there to show you it's not perfect, but you've gone from having these sequences of interest representing a fraction of a percent of the stuff you're sequencing to probably like 60, 70, 80% in some cases. And so it's a massive, massive enrichment. And what this really was, it allowed people to adapt this technology to focus tests for clinical genes. And to show you an example of what happens with this, this is a gene, the COL6A1 gene, that's associated with the congenital muscular dystrophy. We design these baits in green underneath the exons, and you can see the effect of having these baits over here versus not having any baits over there. What it has allowed us to do is say, let's not waste any of our sequencing capacity over here. Let's have it all land here, and not only just here, but very tightly right around the sequences we want to look at. And so it's this development that really led to the clinical use of next-generation sequencing. But the question we had was, you can scale these captures at almost any level. You can scale a capture to 1 exon if you really wanted to. No one really would. You could scale it to a single test. And that was the way that most clinical laboratories in the beginning decided to use this. They said, well, let's make an X-linked intellectual disability panel, a congenital disorders of glycosylation panel. Let's make disease-specific captures that capture just the genes associated with this disease and offer those as next-generation sequencing tests. So that was sort of one end of the spectrum. The other end of the spectrum, which a few big laboratories chose to go with, was let's capture the whole exome. All 20-some thousand human genes, let's capture all of them and look at all of them. And we weren't ready to jump into that business. That's a bioinformatic nightmare, cost nightmare, and many other nightmares. At this end of the spectrum, it was problematic because to follow that approach, every single test you offer is its own test that has to be developed, validated, maintained, and each one probably has a tiny, tiny volume. So if we brought up a test for, let's say, oculocutaneous albinism here at the University of Minnesota, maybe 10, 12 samples a year would come through. We'd be putting a lot of money into developing individual tests that really didn't have enough volume to warrant that. And so the question we asked was, is there a niche somewhere in the middle? And this is where I have to really thank, it's sort of Kenny's idea where he says, well, everyone's going right, let's go left. Let's try out, rather than picking an extreme, which is where everybody was going, let's take the entire test menu of the University of Minnesota, every test that people here send out, and put that as a single test, one single capture. We quickly realized that was a little bit bigger than we wanted to chew off, but we kind of went with that idea. We were funded largely by some of the neurology groups I work with, so we picked a lot of neurology genes and then connected with several other colleagues to develop a 568 gene test menu that covered things like ophthalmology, hearing loss, pediatric genetics, familial cancer, bone marrow transplant conditions, neurology. And the idea was that all of this is one test. We developed one test with one workflow, one validation. So regardless of whether you order oculocutaneous albinism, Fanconi anemia, ataxia, we take the same reagents, put them in the same tube, run the same test. The only difference in this is that bioinformatically, we only analyze and genotype the genes that have been requested by the clinician. And so that's kind of one common question I get is, well, gosh, what if you run into a Fanconi mutation in a muscular dystrophy patient? We're not even looking there. We're not genotyping any genes except the ones that were ordered by the clinician. But this allowed us to develop a platform where we could offer literally 568 genes grouped into about 130 different panels as one test. And essentially in 1 week, we went from offering 6 genes to 568 genes with one test. And so we kind of took a risk with that. We validated this using 20 samples with 37 known mutations, and we picked a lot of tricky mutations, big insertions, deletions. We picked big genomic deletions and tried to challenge ourselves. We identified, of the non-genomic deletions, we identified 33 of 33 mutations. The 4 big deletions of whole gene deletions we've used to develop copy number variation algorithms. And then we validated our ability to consistently get the same results across runs, across technologists, across instruments. So this was about a 3 or 4 year process. It took from me walking up saying, hey, Lisa Scamenti wants me to look at this gene, to we can look at 568 genes clinically, and we can do it reliably, robustly, and accurately. All right, so what's happened in the first year? We first offered this in August of 2012, and I just counted this morning, we received 275 clinical samples, 50 research samples. Again, a lot of this was funded by some of the neurology research groups, like the Bob Allison Ataxia Research Center, and so that's where a lot of that research stuff comes from, is some of the patients we use the same platform. We've completed analysis on about 226 of these clinical samples. And so when you look at, essentially our sequencing volume prior to this was probably 10 to 20 samples per year. So to go to 226 is more than a 10-fold increase in our sequencing sample volume. And it's a nice mix. We've got a lot of single gene tests, which we include in this. It may not be the most effective use of this technology in terms of the power of the technology, but what that allows us to do is drive the volume of this and to keep the turnaround time, the cost down. We have 2 to 5 gene panels, 6 to 10, greater than 10 gene panels. And so the biggest panels we've run have been about 50 different genes. And the bulk of the samples have come from neurology. Again, this was very largely driven towards neurology in the beginning because that's where we were funded from. But what we did essentially was say, using that money, can we get some other people to tag along in this design? And it's actually worked quite well. We've had a lot of good support and feedback from our colleagues in hearing loss, ophthalmology, pediatrics, hem-onc, and BMT. Those are some of the most commonly ordered tests. And this is hopefully one of our successes is our turnaround time. In the very beginning, it was atrocious. I think a lot of our clinicians will agree with that. It was about 16 weeks to get a sample turned around, which reflected the fact, we essentially increased the scale of what we were looking at 100-fold in a week. We just turned on the light switch. We've really, really worked very hard to bring this down over the last year to the point where the average turnaround is just below 8 weeks. And we actually have some new instrumentation that we think is going to significantly bring that down here in the near future. And so we've really done, I think, a lot of work to try to get this process smoother, to get the turnaround time into a range that's helpful for clinicians here. So practical stories how has this helped us out clinically? Story number one, he was actually the very first person we drew. He's one of my patients. And he's a 29-year-old kid who came into clinic with a lifelong history of ataxia. He was petrified. Actually, no, I'll put that in later. He completed a college degree. He was volunteering, so he'd done very well academically. He'd been evaluated previously and just diagnosed with static encephalopathy. But he came to the ataxia clinic because he recently had learned that his cousin, or his father's cousin, had died from acute myelogenous leukemia at age 42. And when they went to the funeral, the local ataxia support group was there. He's like, oh my gosh, there's someone else in the family with ataxia. And this history of leukemia and ataxia, there's a couple things that came to mind. We drew his alpha-fetoprotein. It was elevated. And there's a couple things you think about there. AOA2 and ataxia-telangiectasia are the two you think about. And especially with the family history of leukemia, we're thinking about those, with AT. And so what we did was we sent out the gene sequencing for AOA2, which is the way you rule out that condition. And we sent out the gold standard test for ruling out ataxia-telangiectasia, which is you send a blood sample to UCLA. They irradiate the cells. They look at the cells after a few months and figure out, do the cells heal from this damage or do they die? So it's called colony survival. And they do a Western blot. And I got a call, the Senataxin sequencing was normal. I got a call from the lab at UCLA, and they said, normal, this kid does not have AT. And prior to this, really the diagnostic workup for AT was you do the colony survival. If it's normal, you've ruled out the diagnosis. But we had also decided to sequence this guy for all of these ataxia genes, and we knew when they called, he actually had 2 mutations in the ATM gene for ataxia-telangiectasia. And so previously, we would have never had the resources to be able to look at all these genes, to have this information, and we had them go back and repeat the assay, which the second time around they said, oops, yeah, it's actually abnormal. But they were able to show that the 2 specific mutations we had affected the protein in a way that really didn't make their assay abnormal. So you had a very atypical diagnosis of ataxia-telangiectasia that may have been missed had we not gone in with DNA testing and circumvented in parallel done this with the usual diagnostic workup. I think this has been really enlightening in some cases because we've had some very unexpected patterns of inheritance. And so this was a young man I saw in clinic. I used to work in the bleeding and clotting clinic who had a venous thrombosis after a leg injury. And as part of his evaluation had a borderline protein S level. And it was repeated several times. We said, yeah, we think it's protein S deficiency. And prior to us having this testing, nobody in the Western Hemisphere sequenced these genes for diagnosis. And so we essentially counseled people based on the protein assay. If you had a protein S deficiency, well, that's an autosomal dominant condition with a predisposition to venous thrombosis. So we counseled the family about autosomal dominant inheritance. Well, we actually decided to sequence this guy because he wanted to test some other family members. And what we found, lo and behold, he actually had an autosomal recessive form of protein S deficiency. And so we essentially, based on history and how we've counseled people in the past, assumed everybody with this diagnosis has an autosomal dominant condition. In fact, it turns out not to be true. It's simply because we've never had the ability to look at DNA to actually get to the diagnosis. It's because we were able to that we were able to figure this out for this family. This is an example from the hearing loss clinic of 2 young girls who have what looks like autosomal recessive hearing loss. We had excluded the highest likelihood candidate genes GJB2 and GJB6. And with a lot of conditions, I showed the example with ataxia, once you knock out those few common genes, you often are left with a massive pool of very rare individually conditions. And it's difficult to wade through there and figure out which gene do you look at. And so what the clinician did was sent us both sisters, and this gets to, as you increase the amount of genes you're looking at, it becomes more and more helpful to look at multiple people in the family. And especially if you're going to go to the scale of like an exome, you got to have multiple people in the family to try to figure out, are the mutations or the variants you're seeing segregating with the phenotype or not? And it turned out in this case to be very helpful. We analyzed both sisters. We found more than 200 different variants in both of these sisters. We looked at a whole bunch of genes, but each of them, they both shared 2 variants in this particular gene, one of which was a known pathogenic mutation. The other one we highly suspected, and the fact that it segregated with the phenotype in the family, we were able to make the diagnosis in one quick blood test for what would have been, actually what up to then had been a 5 to 10 year diagnostic odyssey. So those are just pictures of their mutations. And finally, getting back to sort of what's near and dear to my heart, the ataxia. With neurology, essentially genes, and this happens in a lot of other areas, get divided into different areas. You've got genes that you call, well, these are ataxia genes. These are neuropathy genes. These are spastic paraparesis genes. And a lot of that was really necessitated by the fact that we didn't have the resources to look at all these genes in every patient that walks through the door, so we had to try to sort of pigeonhole them into a phenotype and say, well, you look like you have ataxia. It looks like it's recessive. That narrows us in on a reasonable group of candidate genes to look at. And what we did with support from the Bob Allison Ataxia Research Center was say, let's take those blinders off and say, let's test you for every gene we know of that's associated with neurologic disease. I know you came to the ataxia clinic. I know you have ataxia. We're going to look at all these different neurologic diseases. And what we found was, well, gosh, there's an awful lot of variants, about 800 per patient. So this is the participants on the bottom, how many variants we found. So when you take this approach, the limitation is you find a lot of variants. You've got to have good, effective ways to sort through those to separate the wheat from the chaff. But what we ended up finding was the most common cause of ataxia in this cohort of people was actually a non-ataxia gene, quote unquote. It was a gene that had been put into a category called hereditary spastic paraparesis. And no one had ever really looked at this gene before because, well, it was one of these other genes. But if you took off these clinical blinders that we had put on and said, let's just look at all these different neurologic disease genes, we actually were able to get to the answer. And to do this, we had to look at about 250 different genes in these patients, which would have not been practical by prior methods. So where are we going now? I think our future directions, one is we are implementing, probably the Illumina model number doesn't mean much to you, but the Illumina 2500 essentially will allow us, rather than batching samples once every 3 to 4 weeks, to hopefully batch once a week, be able to batch patient samples. So when I said we're going to really be working on the turnaround time, that would chop away probably 2 to 3 weeks off a lot of samples all at once. So we hope to implement that very soon. We, and Dr. Cashton again was talking to me right before the talk, we, based on our experience with these initial rollout tests, said, hey, let's go talk to a lot of other clinicians who might be interested in other testing, including nephrology, cardiology, metabolism, and try to figure out who else is interested in using this platform. And the fruits of that really were, we put together a 1,633 gene testing menu. And I'm very precise about the numbers because the bioinformatics people hate it when you round. So you got exactly. But we've really tried to reach out to a lot of different areas of the University of Minnesota. And I spent the better part of my summer curating, putting together this list. Only last week to have the rug pulled out by Illumina saying that for a fraction of the cost of what we're going to do per sample to put together that 1,600 gene list, we can sequence actually all 4,800 genes that are known to be associated with human phenotypes at a minimum of 20x coverage across every coding base, like 97%, for like peanuts. And I think one of the exciting things about the group we work with is that we're small enough that we're able to really turn on a dime and say, you know, broke my heart after spending my whole summer putting together this list, but shoot, let's try this out. And so I think this is also nice because it validates that initial decision we made when we said, we can pick out little gene panels or whole exomes, or we can do this sort of test menu approach. This is what Illumina bet their chips on. They said, here is a 4,800 gene test menu that you can use to build your institution's test menu. So we are currently piloting this right now, and the idea is that this includes almost all of those 1,633 genes that we put together, plus another 3,200 genes associated with known human phenotypes to put together a test menu. How I'm going to manage that, I don't know. The other thing is it gives an extremely affordable alternative to exome sequencing, to look at essentially the portion of the exome that is associated with human disease. And Baylor just had this paper put out. For those of you who think that sequencing the whole exome will always give you the answer, it did in about a quarter of the patients, but 100% of the answers they got were within that group of 4,800 genes. And so essentially you got no more diagnostic yield by looking at those additional 15,000 genes, probably because we don't understand the function of a lot of those. So there may be a point in the future we look at those, but the idea is not only can we use this platform as the basis of our menu, but it may give us a way of running affordable and maybe more clinically useful exomes. So we're in the process of doing that right now. And I think I'll probably wrap up there with about 5 minutes left to see if there's any questions. 2. Okay. Yeah.
So that's very interesting, very promising that you have now a way to scan for many diseases at the same time. But I was wondering, can you enlist this gene test that you do also for genes that are important for differential therapy? For example, we know that there are many polymorphisms that they predict a different response to anticoagulation or to ACE inhibitors or complement activation. So can we start using that also for differential therapeutic?
I'd say yes, and we've actually brought up and dropped a Coumadin assay 2 or 3 times now. I think twice, because people say, oh yeah, bring it up. But the problem is that people are so used to the way they do things that no one ends up using the test. So these are certainly amenable to that type of thing, but I think there's a lot of stuff that has to happen beyond just us offering it. Is that? Yeah. Okay, so heterozygous deletions, and I assume you mean like large scale, like gene deletions, copy number. Right. So yeah, I kind of danced around that issue a little bit. So I'm like the realtor when you point out the leak. I say, well, let's look at the porch, and we go look somewhere else. But it is for Mendelian diseases, for most of them, the most common things, the most common cause of those diseases are small point mutations you can find by sequencing. But for a lot of these diseases, there's a fairly substantial proportion of mutations that are large genomic deletions or duplications that don't change the underlying DNA sequence that's left. They just remove a copy or sometimes duplicate a copy. So if all you're doing is looking at the sequence, it looks perfectly normal because you're not counting how many copies are there. And so we included in our clinical validation 4 different samples with heterozygous, hemizygous, all different arrangements of copy number variation. And we've been working, and this is some of the money we've used from our grant, to try to develop a robust algorithm to be able to detect those. And the idea is that there's a relationship between the amount of coverage for a given exon. You saw from some of those coverage plots how it goes like this. That is an extremely reproducible plot. So over a given section of DNA, we get a very consistent pattern. And if you have a deletion, what ends up happening is you have half the amount of template there. It ends up reducing the number of reads mapping to that individual area by about half. And so we included several samples with that, and we actually showed we can calculate these ratios and get these solid, like 0.5, 0.49, 0.51. And we can detect these things very solidly. The problem is when we went back and tried to apply this across 11,000 exons to say, well, let's try to minimize the false positives, we ran into a lot of false positive issues we had to try to solve. So we have an algorithm in place. We've used it to call one Fanconi deletion, whole gene deletion of FANC-A. And we've used it to call a couple of other homozygous or hemizygous, which are very easy because essentially you have no reads mapping there in males. We're hoping to roll that out soon, but we also need to figure out how this new 4,800 gene panel plays with that. Yes?
Can you speak to, you said that turnaround time had decreased from 16 weeks to 8 weeks, but if you've got a family with a known mutation, is the turnaround time still 6 weeks?
Right, okay, so in parallel to this whole process, we developed two other, so this is all the next generation sequencing. Once you've identified a mutation in a family, you don't need to do all this. We verify all the pathogenic mutations we find by Sanger sequencing, which means once we've got a mutation, we've got a primer set that can amplify that mutation and detect it, and we've got a positive control. And so from there, if you send us another sample from that family and say, I just want to look for that mutation, all we have to do is grab the positive control, grab that primer set and run it, probably a few weeks. If you're asking about a current sample, probably a few weeks. Indirect way of saying, hey, can you hurry up? All right. Any other questions? All right. Thank you very much. Hey. We've got it. I put it on there. So the sample's there.
I still order it as next gen.
Yeah.
Because it has to go to your lab.
Yeah, if it gets to our lab, we can I mean, that's the main problem.
And that's why I put it.
It was funny
Here's
because
the mom, here's her MR number, here's the mutation.
It was funny because Maggie paged me twice about it, and then Carrie paged me this morning. And so, I was like, I had 3 or 4 clinical people, plus I went to a birthday party this weekend. Someone told me about it. I mean, at the Harper's birthday party. So, hi. Nice to meet you.
Nice to meet you. So, we still have a problem. So cystic fibrosis we're okay with. But,
Yeah.
Ciliary dyskinesia.
Yeah.
Still in that developmental stage.
Yeah.
And how? So we have maybe half dozen families that and they're very interested.
Yep.
And actually, we are too to know what's going on. Does this offer an opportunity if we have these families for any exploratory work, or how do you want to approach that?
Yeah, so we've, I think everyone's always been like, oh, hey, can you test my samples for free? It's like, we're kind of, what we have done is, in the beginning, we wrote a whole bunch of grants for different organizations we worked with, trying to get funding for that type of idea. Because we were kind of looking at how do you grease the wheels and get this thing going.
Right.
And in the end, the only successful one, which really has been, it's been a great partnership, was the Bob Allison ataxia. And that was largely because they're both a research and